Good afternoon, everyone. Today I will present you my PhD research, which was published in Cell Reports a few months ago. In this work, I identified arginine methylation as a novel post-translational modification of huntingtin that is able to regulate its function in axonal transport. Loss of arginine methylation hampers HTT recruitment to vesicles and leads to trafficking effects and neuronal death. On the other hand, the overexpression of the enzyme responsible for HTT methylation, called PRMT6, rescues the effects of mutant HTT on axonal trafficking and cell toxicity. Arginine methylation is catalyzed by a family of enzymes called protein arginine methyltransferases, or PRMTs. PRMTs catalyze the addition of one or two methyl groups to the arginine side chain to produce mono or demethylated arginine, and they are divided in different classes based on the specific reaction they catalyze. Considering that arginine methylation is particularly abundant in neurons and that a proteomic study found some guarantees among HTT interactors, we wondered whether HTT is methylated at arginine residues. To determine whether huntingtin is methylated in vivo, we immunopurified huntingtin from vesicles isolated from the brain of wild-type mice, and we performed a mass spectrometry analysis. We identified the two candidate arginine residues for methylation in the N-terminal portion of huntingtin, namely arginine 101 and arginine 118. With a co-immunoprecipitation assay between HTT and 8 different PRMTs, we found that HTT forms a complex most efficiently with PRMT2 and PRMT6. To determine whether PRMT2 or PRMT6 methylate HTT, we performed an in vitro methylation assay by incubating wild-type or methylation-defective GST HTT with recombinant PRMT2 or PRMT6 and a methyl donor. In our assay, PRMT2 did not modify huntingtin, whereas PRMT6 selectively methylated huntingtin at arginine 118. We next performed a confocal microscopy to understand whether huntingtin and PRMT6 share a subcellular localization in primary neurons, and we found that they co-localized both in the soma and in isolated axons. Moreover, by subcellular fractionation of mouse forebrain lysates, we detected PRMT6 in the fraction enriched in small vesicles together with HTT. This result was further validated by transmission electron microscopy coupled to immunoball staining of purified vesicles, which revealed that almost 40% of HTT positive vesicles also contain PRMT6. To determine whether arginine methylation affects HTT-mediated vesicular transport, we took advantage of a microfluidic chamber in which cortical and striatal neurons connect to each other through microchannels, recapitulating the corticostriatal network that is severely affected in Huntington disease patients. We transduced the cortical neurons with lentiviral vectors expressing mCherry-targeted wild-type or methylation-defective huntingtin with arginine to lysine mutation, and we recorded the trafficking of HTT-positive vesicles. We found that neurons expressing the methylation-defective HTT had nearly 50% fewer anterograde vesicles, a lower linear flow rate, and a shift toward retrograde transport. Very similar results were obtained by silencing PRMT6 with a specific shRNA, demonstrating that HTT scaffolding function in axonal trafficking is influenced by PRMT6. By immunoprecipitation experiment, we found that the loss of arginine methylation does not affect the interaction between huntingtin and molecular motors. However, when we performed a subcellular fractionation from neurons transduced with wild-type or methylation-defective HTT, we observed that loss of arginine methylation reduced HTT recruitment to vesicles by 40%. We then asked whether the alteration in vesicular trafficking correlated with neuronal toxicity and found that overexpression of methylation-defective HTT or PRMT6 silencing reduced neuronal viability by almost 50 and 40% respectively. Through biochemical analysis, we observed that polyglutamine expansion of huntingtin does not prevent the formation of huntingtin-26 complex and the methylation of huntingtin at arginine 118. Therefore, we tested whether stimulating 26 activity could be an effective approach to treat the toxicity and trafficking defects induced by mutant huntingtin. As you can see, the expression of huntingtin with an expanded polyglutamine tract reduced neuronal viability compared to control neurons, which are represented by this dotted line. Interestingly, the TREM26 overexpression rescued neuronal death, but this rescue was prevented by the methylation defective mutant huntingtin. Accordingly, increasing TREM26 expression significantly increased the efficiency of antibody trafficking in neurons expressing mutant huntingtin, but not in those expressing methylation defective mutant huntingtin, indicating that arginine methylation of huntingtin is necessary for TREM26 to improve axonal transport and viability in EHDN neurons. To investigate whether increasing TREM26 expression abrogates mutant huntingtin-induced toxicity in vivo, we crossed HT flies with flies overexpressing TREM26, and we found that expression of expanded HTT completely prevented eclosion, but we observed a full rescue with TREM26 overexpression. To address whether expression of TREM26 rescues axonal transport and neuromuscular junction defects in vivo, we analyzed the fly segmental nerves by confocal microscopy. As you can see here, we found a dramatic increase in the number of satellites and total gluton in ST flies, but this was restored to control levels by TREM26 overexpression. From these observations, we conclude that TREM26 reduces toxicity and rescues axonal transport and neuromuscular junction defects in vivo. And overall, this study indicates that increasing HTT methylation by boosting TREM26 activity could be of therapeutic interest for HT. Thank you for your attention.